invitation. It's great to be able to be here. Really what today is going to be about is, in many ways, just an obsession that I've had now for over a decade. When I was a first-year graduate student, I was kind of thinking about what to do with my life, you know, what lab to join, all these kinds of things. And while all of this was going on, I actually picked up a New York Times and I read a statement that basically changed my life, and that's this one right here. So if you remember, they had just sequenced the second genome. It was Mycoplasma genitalium, okay, so Haemophilus influenza and then Mycoplasma genitalium. And that had enabled them to do what you guys will remember at the time is incredibly primitive, but the first comparative genomics study, right? So they took the two genomes, they looked at the list of genes in each genome, and then they just took the intersection of that, and they said that's the minimal set of genes required to sustain life. You remember that? Of course, we've come a long way since then, but at the time, for me, as a first-year graduate student, just completely riveted me. I couldn't believe it. We're sequencing genomes. We're starting to talk about designing minimal cells. So I'm just reading this article, absolutely enthralled by it, and then I come to the end part where they're talking about actually one day making a cell that has kind of a minimal gene component. And Clyde Hutchison, who's with the Venture Institute, then comes up with this quote: "The ultimate test of understanding a cell, more than being able to build one, would be to make a computer model of a cell." Because that implies a fundamental level of understanding. And literally, I think about this quote probably every single day. So I've thought about it every day, certainly since I've read it. And I've just been totally intrigued by this idea that maybe we could get to this point where we're really having these comprehensive models of cells. And of course, to this crowd, it's not going to be any big surprise what you could do with a model like that. Just our ability to go in and actually interpret the large-scale data sets that we're now able to generate so quickly and relatively inexpensively. Our ability to predict complex behaviors. As many of you know, I mean, we read all the time in the newspaper about the cancer gene discovered or another Alzheimer's gene discovered or something. But of course, cancer is not a one-gene problem, right? It's the interaction of thousands of factors. And so can we make models that would enable us to predict complex diseases, complex behaviors? Of course, I just talked about designing organisms. As we start to look at trying to make, let's say, antibiotics or get more into bioremediation or other biotechnology efforts, we'll want to make sure that we can do those things without side effects. And so to be able to do that, to make them safe and efficient, we'll need computer-aided design tools just like we do in the other engineering disciplines. And of course, all of this with the maximum possible efficiency. So really what I want to do today is just pose a few questions, show how we've come in kind of addressing, trying to get at the answers to those questions. And so I'll just start with the first one, which is, can we build whole cell models? Okay? So we went in with this idea that we really wanted to try to get as close as we could to having a comprehensive model of a cell, full life cycle, taking all of the genes and their annotated functions and all of the molecules into account. And starting from when I first read that article, okay, a lot has happened. And the key thing that I want to note, I can't go into all of the details here of all the different methods and models that have been made, but I want to note one thing, which is that it wasn't just whole cell modeling is just not just an effort of modeling approach. Okay? For whole cell modeling to occur, it's clear that we need advances in data generation, data interpretation. So you see here, there are groups that have made massive experimental efforts that have contributed to this, machine learning and kind of inference methods, databasing. So all kinds of different resources and skill sets have had to come together and will have to come together in this kind of an effort. I felt like just looking at the modeling, when we first started thinking we could actually do something like this, which was about 4.5 years ago, I felt like there were two major lessons that I had learned from the last decade. And the first of them was that whole cell modeling requires a variety of approaches. So there's no single method that's sufficient to model whole cell, whether it's ordinary differential equations, some stochastic simulation, flux balance analysis. None of these is sufficient by itself. And the reason is because of the way that we understand these different processes. Some processes we understand in such great detail, while others we hardly understand at all. And so trying to reconcile different data sets and different levels of understanding, it's a really fundamental challenge that would preclude, in many cases, one modeling approach or another if you felt like you had to use only one. And so the corollary to that is that instead of focusing on a novel modeling approach, why not focus on the integration of different modeling approaches? Okay? And so that's really what we decided to do in going about this. So we've done some work I've done some work in this area before. My PhD thesis was largely about trying to integrate metabolic networks using flux analysis with transcriptional regulatory networks. We came up with ways to integrate that together with ODEs. So we had a little bit of background, but even at that time, I never considered doing anything at a truly large scale. But now I want to, but it is similar in concept, and so I want to give you an idea. This is just showing very schematically what we've done, and I'll go into more detail. But basically what we do, we take the whole functionality of a cell, okay, and we pull that apart into modules that we consider to be somewhat independent of the others. So I'm just showing 8 here. Like metabolism, for example, being somewhat different than protein complexation, okay, or DNA replication. So we pull these things apart into modules. Now, for each module, we are going to make a model of that individual process. And because we've pulled them apart, we can treat each one uniquely. Okay, so we can go in and we can say, okay, let's just look at what the data is that's associated with DNA replication, right, or decay. What data do we have? What does that data look like? Is it qualitative or quantitative? Do we associate it with fold changes? Like how do we understand the data that we have, and how can we interpret it? And then furthermore, if you just look at like the reviews of those processes, what does that look like? How do we understand this process? Do we understand its time evolution? Is it just a series of interactions? How do we put that together? So taking all of that data and all of our understanding together, we then go in and choose the best possible modeling approach for each model. Okay? This gives us a lot of freedom because we can just, in a very objective way, we can try to just hone in on the best way to model that. If models existed, we always favored those, right? We tried to just really favor what had been done before. But of course, it's a challenge. Even the data sets themselves are not always internally consistent. And so it was a very interesting challenge to try and bring all of this together. Now, I told you that we separate these models, and of course, that is what we have to do. But in fact, they're extremely integrated. Okay, and I'm just showing you some of the arrows here, but I want to give you this sense that these models are constantly talking to each other, like in reality, in a cell. All of these processes are constantly talking to each other, and that's part of the advantage of a whole cell model in the first place. We want to be able to capture those interactions. But it's so complicated that it really makes it difficult to have a unified, integrated model that takes care of all those processes. And so what we did is come up with a strategy that, often I ask the crowd how many have coded a Runge-Kutta integrator. I think in this crowd I'd probably have more than in some of the other groups that I talk to. If you've coded a numerical integrator, then it's very similar to what we're doing here. So what we assume, we make this assumption that even though these processes are all integrated, connected, interacting, okay, we assume that at a short enough time scale, okay, and in our case we just say less than a second. So within a second time scales, we're going to assume that those processes act independently. So they're not independent across a second, anything longer, but within that second, they're independent. What that enables us to do is when we run a second of simulation time, we can simply run every module individually. And right now, we even do it in series. We just randomize the order in which we run the module, and we run them in series. We want to parallelize that. But we run that second of simulation time, and then all of those modules they're acting on this large, massively heterogeneous data set, okay, that includes, you know, the number of every complex of a certain type, as well as where exactly on the chromosome every DNA binding protein is bound, okay? So we have all of this data, and so at the beginning of the second of simulation time, it has a value, let's say, at time step I. Then we run the simulations. They all operate on this at time step I, and the output of all of those modules together gives you the value of the data set at I plus 1. Okay, so that's essentially, I'm oversimplifying a little bit. Happy to talk about the details later. We took this method and we applied it to Mycoplasma genitalium, maybe in part because that's what I first read the newspaper article about, certainly in part because it's the smallest genome of an organism where we could still culture it. Okay, so we culture this in the lab. We have a kind of an active area where we're culturing mycoplasma. So it's got the small number of genes. It's annotated. There's a lot of homology to E. coli, which was important because a lot of the things that we know come from the E. coli data, and then we can grow it. This came from a review of our work in Nature Biotech, but I liked it a lot because it kind of, again, looks a little bit like how I would draw a numerical integrator. So what we're doing is we're taking this large, like I said, heterogeneous data set, and here they've grouped it for us into a number of different data types. Okay? Then we have these 28 modules, so not the 8 that I just showed schematically, but now we have 28 different modules, okay? All kinds of different things. DNA damage, DNA repair, FTSZ, that's a ring for cytokinesis, so polymerization of the ring. And each of these we model with its own mathematical process. And we just run through this whole process through each module at each time step until the cell divides. So now, this is actually just as exciting for me as anybody else probably, but some of my students recently put together something called Wholesale Viz, which is just, so for us to run one simulation of one cell dividing one time, that generates half a gig of data. It's very interesting when you compare, you know, the genome, of course, is barely a JPEG worth. So let's see here. So what I want you to do, we're just starting to visualize it. Like actually looking at this data has been really challenging. So here I'm just going to show you some of the views. We have 30 or 40 different views, but I want you to look here. You're going to see the cell growing and dividing. Here, this is a metabolic network, glycolysis down here. This is nucleotide scavenging. The size of the circle gives you an idea of the concentration of the metabolite, and the arrows are going to get thicker or thinner. Okay? Here, it just shows you the relative expression of everything in the cell from RNA to protein to complex. Yellow means there's a lot of it. Blue means there's not so much. This is one of my favorites. So you're going to see the chromosome. This is the chromosome, and every line on here is something that's attached to the chromosome, okay, on either strand. And you'll see as it replicates, you'll see something happen here. Here, this is a linearized chromosome where you can actually see this is the DNA A binding complex, opening up the chromosome for the holoenzyme, and then this is replication down here. And here you can see all the different translations that's occurring on available mRNAs. So this is like a gene list, okay, going through the genome. So you can go through and kind of see how this looks. Like I said, I've only seen it a few times, so that's kind of awesome for me to see there. It just opened up. Okay, it's making it, changes all the DNA binding. Yeah, it's been a lot of fun to see. And then over here in a second, you'll see this thing pinching as the ring forms. And you get replication of the cells. So a lot of data in there. And like I said, we're just starting to look at it. So let's go to the second question, which is, we've spent a lot of time talking about, well, if you had a model of a cell, like what would you actually be able to do with it? And the first thing you want to do is be able to verify that you're capturing the critical information that we have about this cell. So we've now started to just compare this to all the data sets that exist, both some that we used to train the model with and some that we didn't. And the first thing that we thought we had to do, like the cell had to be able to actually double, okay? And it had to be able to divide. And more particularly, we wanted it to do that at a time scale that was comparable to what we measured experimentally. So I'm just going to show these briefly, but we went through experimentally and measured the doubling time of mycoplasma, and we used that as a key parameter that we fit to try and get this model working. We didn't have to fit too much, but there were a couple things. Again, more than happy to talk about it. Like I said, every one of our simulations is a single cell, and so we had to run hundreds. We've now run thousands of simulations. I should note, every simulation, all the source code, every single thing we did is available for free at simtk.org. Open source, open everything. There's even a little tutorial on how to add a module for those of you who want to make a better model. So we ran each of these, and here's a time course for each one, and so we got a median doubling time that was roughly the same, so that was nice. We also looked at the cell composition. We want to make sure that that was relatively similar: DNA, lipid, protein, and RNA. So here you can see in black in blue the simulation, and in black the experimental measurements. I do want to make one note here. We often are faced with this problem with this data that's not necessarily internally consistent. In this particular case, what's interesting is, we had a measurement for DNA content that came from the 50s or 60s, where someone had dried out mycoplasma cells, cracked them open and figured out their relative content. These days, we don't actually, that measurement can be superseded by the genome, right? So the genome sequence, we can just add up molecular weights, and we can also get a rough estimate of how much DNA we'd expect to be in the cell. What's interesting about those two measurements is they don't add up at all. So in fact, what was measured back in the 50s doesn't even give you a fifth of the chromosome we would get from the genome sequence. And so there's this real challenge in trying to just integrate all of these disparate data sets and somehow make them reconcile with one another. We then wanted to see that every component of the biomass also was able to double, and so we were able to make that happen. Just notice here briefly that the trajectories are somewhat different. Like RNA, because of its low cell count or low copy count, is a much noisier trajectory to doubling than some of the others. Okay. Another thing that we went in and looked at, I'll show you here. Okay. Take a look at this one. So here what we're looking at is the production of all the RNAs, protein monomers, that then mature RNAs, and then the protein monomers themselves, mature protein monomers. What I want you to see, again, blue is low and yellow is high. So there's typically a lot less RNA than protein in these cells, and of course that's because the half-life of the RNA is so short with respect to the protein. In looking at this, we found, just looking in more detail here, this is just kind of a fun genome view. I'm mostly just showing it for my own enjoyment. But I want you to see we can actually look at a detailed time trajectory for each one as well. And I want you to look here briefly at the transcription unit 6. Okay, so basically here you can see, for 6, you can see the mature RNA. Here the monomers are being made, and the dimer, of course, much more stable. These are binding to make this. And then you can even see the flux going through this enzyme once it's formed. But what we saw, and what I was very encouraged by, at the time I was shocked by, because I was not familiar, this was kind of early days, and I hadn't seen some of Sunny Shi's fantastic work in this area. But you see the burst-like expression. So you get, because of the short half-life of the RNA, you just get these little bursts of mRNA formed, and then you get bursts, basically, of monomer and subsequently dimer formed as a result. So this was very interesting for us to see at the time, and we were also very excited then when Sunny Shi's stuff came out and they started showing, he was able to, using a YFP fusion library, he was able to count the numbers of protein and the numbers of mRNA using mRNA fish in the same cell. And they found, basically they found that there was not a correlation truly between the mRNA and the protein, again because of that short half-life in the individual cell. So each one of these points is an individual cell and how much mRNA and protein they counted. What we saw, we can't compare directly, they were doing their work in E. coli, but we saw similar distributions of both the protein and mRNA, and also a lack of correlation, and also this burst-like expression. So that was a nice thing for us to see as far as validating. And you can see the burst here as well. So now we've compared our work in addition to some of our own experiments in the lab, which I'm not going to report on today, we've been able to compare it to the transcriptome, compared it to the fluxome, we've compared it to metabolite concentrations. We've compared it really to as extensive a heterogeneous data set in particular as you could. We've compared it to everything we could possibly compare it to that's published. And it looks pretty good. You know, there's definitely always room for improvement, but I'm an engineer, so I was happy enough to start then moving forward with it. And so the final question that I want to talk about a little bit is, can we predict novel behaviors? Can we really get in and find something that we had no clue about, that we had no access to? And we're doing a lot of this lately, particularly in the lab, but I just want to show you one thing briefly that I thought was a very exciting, complex behavior. So here, what you're seeing, again, this ring is supposed to be the chromosome, right? Here's the origin at the top. And what you can see in this plot is basically where different proteins are binding. So if you look at this outer blue ring, you can see where all of the proteins bind. And you see that the chromosome is really quite heavily visited. And in fact, most of the chromosome space is visited in one way or another after really within the first 30 minutes of the simulation. You can also look at the RNA polymerase here, the purple ring. You can see genes that, like this one, for example, is almost definitely like an rRNA gene or something that's really highly expressed. DNA is interesting. DNA is a complex that forms at the top, at the origin, and it opens up the DNA so that the holoenzyme can get in there and start replicating. If you were able to zoom in here, you'd see that you can actually see the boxes where it has to bind. There are 4 binding sites that it uses. But there are also lots of buffer sites, so it binds all over the chromosome, and so you can get an idea of where it's binding. But this is the one I want to focus on because it was a really surprising test at the time, which is that DNA polymerase travels very quickly. So it has this lighter green, which means that it's not spending much time here, right? So it travels very quickly in the beginning of replication, but then it starts to slow down, kind of slows down through the rest of replication. So we looked at that, and we had no clue what was going on there. I'll show you here. Imagine that I'm taking this circle, okay? I'm making a cut, okay? And I'm just opening it up. So now you've got the linear chromosome, but we can look at who's visiting it over time, okay? So here's the origin, here's the terminus. And now you're going across time and seeing where everything binds. And I'm showing you those same 3 proteins, or protein complexes. So the DNA A, the RNA polymerase, and the DNA polymerase. So here you can see the formation of that unit, right? Here you can see transcription of all the genes. It's really fun. We can see, like, for example, you see all the slopes because go in this direction. This is thought to be so that the replication won't bump into the RNA polymerases, right, when it's traveling along. You can also see, you know, for example, you can read from the vertical, you can read the gene length, you can tell how fast it's transcribing that. Here's something that's transcribed really heavily. If we were to really sit and stare at this for a long time, I could show you where collisions happen, and I could also show you things like this jagged edge where you see kind of the Okazaki fragments, et cetera. So it's a very fun graph, but the key thing, again, that I want you to see is that this is going very quickly, right? So here, it's traveling very rapidly, and then it slows down for the rest of DNA replication. So that was an interesting mystery. And then as we were continuing to kind of think about this, we noticed another thing that kind of struck us as being very unusual and interesting. And what that is, like I told you, every single cell is different. So there's definitely some stochasticity in the model. So a different cell cycle that we simulate will give you a different answer, so to speak, a different half gig of material. One thing that we noticed was that we had this variation of the whole cycle time. Okay? We had this variation around that time. And you can see what it looks like here. But if you look at, well, let me just tell you. So you can divide that cell cycle into 3 major phases. One is a phase of replication initiation. That's when, that's right here, when the DNA is binding to the top of the chromosome. Then there's replication itself. And then finally, after replication is finished, you have cytokinesis. So if you look at cytokinesis, the time that it takes to go through that is very well conserved, and it's always kind of about an hour. But look at this distribution here for replication and replication initiation. Very surprising, to me at least, at the time, to see that these things had such a wide distribution, and yet they all contributed to this cell cycle that had a much smaller distribution. We didn't code anything in here that seemed to have any relevance to that at all. And so we wondered what was going on with that. I'm going to show you quickly how we figured it out using one of these. Okay. So there's a lot of fun stuff here. You can see the super helicity of the chromosome. You can see the shape. Here you'll see the contractile ring. These are fun, but kind of ignore them for right now. So here you're going to see the building up of the DNA A complex that opens up the DNA. Okay, this turns out to be a 29-mer the way it's modeled. This is a linear version of the chromosome. What you're going to see, this is count of all the DNTPs, the DNTP pool in the cell. So what you're going to see is that this goes up, okay? This has a trajectory that you can see, and you'll just watch it for a second, see what you think. Okay, so this complex is being built. Boom, once it forms, this thing shoots down, okay, and you have that fast replication of the chromosome. Okay, hopefully you saw that. Maybe I'll just stick around for a second because it is fun to watch this ring being formed. Okay, so now you're going to see the ring. It's going to keep going down, and then eventually it's going to fade out. Okay. We'll go there. But watching stuff like this basically started to give us this idea. So here, if you divide things into replication initiation, replication cytokinesis, okay, you can divide these things. And so, so what I want you to see is basically that as the DNA A molecules are making a complex, okay, it builds up, it builds up, it builds up. You can see it's a little bit stochastic here. Then what happens is as soon as it hits that complex, boom, replication goes really fast, and boom, the DNTP concentration floors out. Okay, you see that? So what that told us is that when the DNA A is building up, the DNTPs aren't being used yet. Okay, so they're forming a little pool in the cell. And once the DNA A makes the complex, all of that pool is used, so you have a surplus of DNTP. And so that holoenzyme complex, it can just fly along as fast as it can possibly go. Okay? But once the pool is exhausted, okay, then it becomes resource limited. Okay, so it has to slow down, and it can only bring in the DNTPs at the rate that they're being produced. And so that's what gives you these two phases. And what was so exciting to me about that is it just suggests this totally emergent control. Okay? And what it means is that, what we in fact saw, that even though you have a lot of variation in the replication initiation duration and the replication duration itself, they're inversely correlated. High duration of initiation leads to low duration of replication because it can go faster for a longer period of time. So to me, this is just one. We have several of these interesting stories, but this is just one very compelling story of just the idea that once you start to try and put everything together in a model like this, it takes on a little bit of a life of its own, right? So that was almost done. So I'll just go back to the questions. So can we build a wholesale model today? So we've made some progress. Very excited about it. Our ability to capture some of these system-level phenotypes has been very encouraging. Like I said, we're doing a lot more of this in the lab, and I believe that it will enable us to predict novel behaviors in the future. So with that, I'll just close quickly. I want to thank my lab. To get the theoreticians started in the lab, we always do a cooking class in San Francisco. So I want to show you Jonathan and Jay Adida here. These are the two main authors who worked on this. I think they're making a chocolate mousse. And then I also definitely want to thank the NIH really being ready to support a crazy idea, and we could not have done it without them. So thanks, and I'll take any questions.
Thanks. Okay, we have time for a few questions. And could the next speaker also come up now so we can let's get started? Great. So, yes.
Yeah, thank you for a fascinating talk.
Thanks.
Obviously, there are many details that you couldn't tell us because of time, but I have two questions about, for example, the pinching off. Are you modeling the bilayer or the details of the membrane, or you are saying at some point there is an event that triggers the pinch off? And the second question, and I'll take the answers, is do you have a systematic way of thinking how to validate some of these things in a way? I'm sure that you might have seen things that are not working, and so those will be as revealing for your model as those that are working.
Yes. So thank you very much. Very interesting question. So the first one, is how do we incorporate the model of the pinching of the cell? So what we model specifically, there's been a model of FTSZ polymerization, okay, polymerization, which we pretty much took straight from the literature. You basically are making these longer polymers of FTSZ. And then when they constrict, Grant Jensen and others made a model of that constriction. So this is one of the cases where we really relied very much on existing literature. Now, does it gather the... it doesn't explicitly gather the membrane around itself, if that was what you're asking. But it does kind of make a small ring that then pinches, we assume, is kind of the inner ring that pinches down until it's closed. The second question, and I hope that's... I can't give the full detail, but we could... Let's call that a starting point and talk after. So the second question, which is... how to validate or how we go about it systematically. Yes, so definitely, I get much more excited when the model's wrong than when it's right, actually. And the reason is because if the model's right now, I just assume, okay, well, that means by and large we understand this part of the network relatively well. Whereas if it's wrong, that's like a big opportunity for us to learn some new things. The most exciting work we've done in this area, this is now where we're focusing a lot of our lab work. So we've recently been looking in particular at how knockout mutant phenotypes have been predicted. Some of them are right and some of them are wrong. The wrong ones have pointed us to some really exciting discoveries because the model can actually kind of tell us why it's wrong and suggest new experiments to do. Yeah.
Andrea?
So a couple of questions as well. So the first one is still in vein of the validation. So you showed some novel behavior that has been predicted by your model, but I didn't see the experimental validation of that. So is that because you always have novel behavior? That doesn't mean that.
Yeah,
that you
it doesn't mean it's correct.
Right.
Definitely.
So did you actually check it out?
So our biggest challenge. So I guess I'll divide this into two questions. So are we currently pursuing validation of the model? So yes, I didn't show it just for time, and it would take a while to set up, but we've done several now instances of kind of independent experimental validations. This case is a little tricky. Our biggest problem with this model is really that it was done in Mycoplasma genitalium. It's a pain to grow, right? Even just to grow up a mutant cell takes months. So in this particular case, we've started trying to address this by labeling the DNA with fluorescence, and we're kind of trying to look at distributions of how much fluorescence is in the different cells. The results are interesting. I won't say that it's very hard just to be able to look in the same detail that we can in the model. So we're getting there, but it is, in this particular case, the validation is tough. But I will say we're pursuing it much more actively in places where we can get at mycoplasma in more detail.
And the second question is that I assume your model offers a number of opportunities for exploring what is the range of scale over which different phenomena are coupled in the cells. So you have made the simplifying assumption that everything below 1 second would be uncoupled, everything above 1 second would be coupled. But in practice, that is completely different from the different compartments. So have you tried to see whether I assume, for instance, that protein complexation and signaling are much more coupled at the sub-cell level than, for instance, transcriptional regulation, which takes 30 minutes in any case to do anything.
You're right, and I oversimplified a little bit, as I mentioned. There are some places where they're extremely tightly coupled. The big ones in this model are energy, right? So almost every module, every submodel requires energy, and in many cases, if you gave whatever, let's say you took one module and you just said, okay, take whatever you want, it would take all. So you have to be very careful with that. So we do have some cases. So energy is the big one, and the other big one is just there's the chemical integration of the energy, and then there's the physical integration of the DNA binding proteins on the chromosome. Like, it really is kind of a molecular ecosystem. It's astounding how many collisions occur, and handling that is another great challenge for trying to trying to put these different modules together. So yeah, even though I gave this over...
I guess what I'm asking is, are there tunable parameters, for instance, like what is the decoupling time of a particular module, such that you can change it, nothing happens, and then there is a critical sort of fall off the cliff type barrier at which the behavior of the entire system is compromised because you're exceeding.
Yeah.
So
that's a great question, actually. So I hadn't thought about it that way. But so we certainly are aware of the things that kind of required special treatment. But it would be nice to try and come up with a more rigorous defense of why we did it, because mostly we just did it from from what we observed, not from any rigorous way. So yeah, thanks.
So I think we need to move on now. Sorry. So let's thank Markus again for a really terrific talk.